Okay, without further ado, I will introduce our phenomenal speaker for today, Dr. Amy Rowat. Amy is an associate professor of integrative biology and physiology at UCLA. She's also a GFI research grant recipient, and we're so proud and honored to have her as part of our program. She is a member of the Cancer Center at UCLA, the Jonsson Cancer Center, the Broad Stem Cell Research Center, the Bioengineering Department, the Center for Biological Physics, and the Business of Science Center. So hopefully that gives you an idea of the range of expertise Amy demonstrates and is bringing to us today in her seminar. My favorite fact about Amy is that she holds degrees in physics, Asian studies, French, mathematics, and chemistry. So I think she just is incredibly effective at bridging kind of the discourse across all of these different disciplines, which lead to really interesting intersections and really interesting perspectives as part of her research. So Amy, we're so honored to have you here with us today. You also have the best name out there, so it's an honor to introduce you. And without further ado, yeah, Amy, you can take it away. Thank you.
Thank you so much for the warm introduction, Amy, and to you and your team for putting together this amazing seminar series. So I'm delighted to be able to talk to you today about some of our new and emerging results on cultured marbled meat. I'm a biophysicist, as you've learned, and so I'm going to spend the first part of my talk telling you a bit about my background, what our lab has been doing, and sort of our core activities in cellular mechanobiology until we got into the cultured meat space just about 2 years ago. So we think about cells as materials, and I've shown here one of my favorite pictures of a red blood cell and the beautiful shape transition that red blood cells undergo when they need to transit through small capillaries in our bodies. You can see here, this is the direction of flow in the direction of my cursor, and they adapt this umbrella shape that enables them to have minimum resistance to the fluid flow, but they have such amazing properties as materials that when they come out of that confined space, they're able to spring back to their original discoid shape, which enables optimal surface area to volume for exchange of gases, which is a major function of the red blood cell. So this is something that really captivated me as a graduate student in biophysics and has really sort of provided the motivation for a lot of our research investigating the relationship between structure and function and thinking of cells as materials. So we've been focusing on understanding cells as materials, particularly in the context of diseases like cancer, where we know that cells need to deform through very narrow gaps, and that enables them to spread through the body. This is also true, of course, of immune cells as well. So how cells deform in response to these physical forces, how they might respond to the physical forces of fluid flow, fluid shear stresses, as well as other mechanical cues such as the stiffness of the surrounding extracellular matrix or scaffolds that they can adhere to, they can attach to, we know also helps to regulate the material properties of cells. So all of these properties of cells play a role in various different physiological and disease contexts. And I've shown a few of them here at the bottom of the slide, ranging from metastasis and drug resistance, of course, differentiation, tissue morphogenesis, as well as a host of various other diseases and physiological functions such as proliferation and immune response. So just to give you an overview of some of the main goals of my group, we're really focused on developing new technologies to measure the material properties of cells and to address the current gap in the throughput of these techniques. We're building these higher throughput platforms to enable us to measure the mechanical properties of cells across hundreds and thousands of samples. And this is really important to be able to develop a systems level understanding of the mechanome. What are those molecules and pathways that are regulating cells as materials? We're also working to define molecular mediators and pathways that determine how cells regulate their material or physical properties. And this can be in response to soluble cues, such as factors that might be in cell media or in physiological cues in circulation, as well as physical cues such as the stiffness of the underlying matrix. And finally, we're very focused on translating our discoveries from applications from cancer to also cultured meat. Of course, the cultured meat is going to be the focus of my talk today, but I'm first going to give you an overview of just a couple of main themes of research in our lab to give you a sense of our core expertise and some of the techniques and tools we use and have been developing. So one major question is, what are those genes and pathways that regulate cell material properties? Another way of thinking about that is like, what are the factors that regulate cellular texture? So to address this question in a higher throughput way, we developed this method that relies on cell filtration. So we have cells that we push through these porous membranes using applied air pressure. And the amount of the cell suspension that filters through depends on how deformable the cells are. If those cells are stiffer, they block or occlude the pores in the membrane and prevent the fluid and the rest of the cells from flowing through. So this can result in reduced amount of filtrate in the bottom well or increased retention in the top well. So this very simple, straightforward readout has enabled us to scale up the process. It's very analogous to bubble tea, just to throw in a food analogy there, where those little tapioca balls can get stuck at the end of your straw unless you apply enough pressure to be able to deform it through the straw. So by measuring the volume of filtrate, simply using a plate reader, we're able to scale up this assay so we can start to assay hundreds of cells across hundreds of different conditions across multi-well plates. And this enables us to compare and identify factors that cause cells to be more or less deformable. So we recently conducted a screen on cancer cells. We know that more invasive or deadly cancer cells tend to be more deformable than the less invasive, less deadly cancer cells. So we identified small molecules that cause those invasive cancer cells to be stiffer or less deformable. And this just gives you a snapshot of the types of classes of molecules that we're identifying. The extracellular matrix and cytoskeletal regulating genes, protein or targets, molecules related to regulation of ECM and cytoskeletal proteins are found in the top category. The darkest red color shows you the most statistically significant hit. But we find a host of other types of molecules regulating cellular deformability as well. So this is enabling us to merge these findings and to perform some meta-analysis using bioinformatics and techniques and pathway analysis to predict some key drivers of mechanotype, of cellular mechanotype. So overall, we're excited to be able to dive deeper into to develop a more systems level understanding of the types of molecules and pathways regulating how deformable cells are. So honing in on more specific pathways, we've also been investigating the effect of soluble cues on cellular mechanotype as well, and specifically focusing on stress hormones, which we know are really critical for fight or flight response. Stress hormones like epinephrine and norepinephrine are circulating through the body and have been shown to play an important role in driving cancer progression as well. So we know that these hormones act through this beta adrenoceptor, a G-coupled protein receptor in the plasma membrane, can be activated by the agonist isoproterenol. So over the years, we've found that the isoproterenol activation of this beta adrenergic signaling pathway makes cells stiffer or less deformable, as we've shown using our filtration technology as well as atomic force microscopy. We also know that at the subcellular level, we get increased F-actin in the cortex of cells in a suspended state, and also remodeling of the actin cytoskeleton in cells in an adhered state. And finally, we've shown that this soluble stress hormone cue actually increases the amount of forces that cancer cells are exerting on their the substrate. And this we can show by this, we've shown through this micropillar assay where cells attach and pull on these pillars, and we can measure the displacement of these pillars that have a known stiffness to be able to back out the forces that cells are exerting. And this is showing that the activation using isoproterenol of beta adrenergic signaling pathways increases cellular force generation. So that gives you a sense of how the intersection of these soluble cues in the cellular environment together with physical factors can be very important in determining cell behaviors. We know that these soluble cues, such as these stress hormones, can be translated into physical forces, and we've elucidated the pathway through which that's happening, which I won't go into detail in today's talk. But we also have preliminary data showing that the stiffness of the underlying matrix can regulate the response of cells to stress hormones. So this really highlights this bidirectional feedback between cells and their underlying matrix, and how soluble cues can regulate those behaviors, and vice versa, how the cell matrix interactions can potentially impact the response, how the cells respond to the soluble cues. So that gives you a snapshot, sort of bird's eye view of some of the research projects ongoing in the group. And I've been, of course, another major part of my role as a professor is to teach students and engage with the public. I'm at UCLA, a public institution, one of the country's largest, and so to be able to engage students in learning science, I've been working for many years to use food as a tool to communicate sophisticated concepts in science. And this work really started when I was a postdoc at Harvard and developed the first science and cooking class together with chefs to be able to provide a more accessible way to engage students in learning physics. So since then, I've founded this organization at UCLA, Science and Food, and teach an undergraduate course on the topic each spring. We also host public events to promote public dialogue around science and food and science. And I also lead a team of undergraduates who've been working on developing high school curriculum on this topic, which we recently piloted in a summer enrichment program online for Los Angeles Unified School District this past spring. So one of the really exciting aspects of using food to communicate science is that food provides really accessible ways and examples of science. So for example, shown here on the bottom left, you can see these cubes of Jell-O, which we discuss in the class, how the gelatin molecules are cross-linked together to form these elastic materials. And the students can actually do hands-on activities in their lab and in their homes to be able to measure the elastic modulus or stiffness of the Jell-O cubes. We then relate how the texture relates to meat, for example, and discuss down to the molecular detail of some of the cross-linked actin proteins that are also important in meat texture as well. So some of our other work, we've shown that integrating taste tests into the classroom can actually enhance student engagement in science, and so this is just another example of how food can provide a very effective medium for science education. So in addition to my work with science and food, I lead the Eat Well pod of the UCLA Semel Healthy Campus Initiative Center. And the mission there is to promote knowledge and access to both healthful and sustainable food choices. So our group spans both academics across all disciplines and also operations as well. And we work closely with them as they roll out, for example, plant-based burgers into the dining halls at UCLA. And we can sort of partner that with more education about some of the environmental aspects of meat and plant-based meat. Finally, I also lead a NSF training grant, Innovations at the Nexus of Food, Energy, and Water Systems. That's been running for a couple of years now, training graduate students on an integrative understanding of food, energy, and water systems. So with that background now, my expertise in cellular mechanobiology together with food and science, along with the underlying sort of increasingly urgent needs in society right now, from a decrease in environmental resources to this extreme demand for alternative protein sources, this really set the stage for us to be able to merge our interests in cellular mechanobiology and food. And this was really catalyzed by graduate student Stephanie Koveski, who joined the lab just over a couple of years ago, and really set forth on our cultured meat research program. And we're extremely grateful to be supported by wonderful partners like the Good Food Institute, who've supported us both financially, but also in creating this amazing community of researchers that really helps to advance the field. Of course, New Harvest and also our NSF Infuse training grant have been excellent partners as well. So the main question, which challenges many of us in the field, is really how can we produce cultured meat that's both cost effective and has minimum environmental impact? And it turns out that there's a lot of cellular mechanobiology that goes into cultured meat production. So when you think about cells, culturing cells in a bioreactor context, cells typically need to be adhered to some matrix. They could be adhered to each other, but typically there's some sort of microcarrier particle. And so those cell matrix interactions become really critical. Of course, how the additives in the media impact cells also depends on the cell matrix interactions and vice versa. Of course, there's physical forces due to exchange of media and fluids to be able to provide nutrients for the cells and remove waste products. So this fluid flow also creates physical forces that can impact cell behavior. And all of these factors together, we know, regulate very critical behaviors that are important in cultured meat production. We need cells to grow, proliferate. We need them to differentiate, for example, to produce muscle fibers. We also need to optimize for production of proteins and accumulation of lipids. And all of these outputs are ultimately what's important for the texture of cultured meat. So I'm very grateful to be working on the mechanobiology of cultured meat with a very talented team of both postdoc Sam Norris, as well as graduate students Stephanie Koveski, New Harvest Fellow, and Kathleen Chen, an Infuse Fellow. Talented undergraduates are Ashton Davis and Kian Amati, and we interact with all the other members of our group who are working on similar concepts in other systems. So of course, we're also grateful for our team of collaborators, at UCLA that span psychology, environmental sciences, adipocyte biology. This is a very multidisciplinary field and requires crosstalk across disciplines. And of course, our funding support as well. So, so with that, I'll launch into the segue now on our research in cultured meat. And this really is founded in the exciting potential that in cultured meat has to reduce the environmental impact of what we eat. And there's increasing evidence that's showing the significant environmental burdens of conventional meat production in terms of greenhouse gas emissions, land use, antibiotic resistance, etc. That's really been highlighted, I think, also through the COVID pandemic. Now, there are, of course, varying views out there, and the method of livestock agriculture really is determining some of the environmental burdens. Work from Jason Roundtree, for example, shows that regenerative agriculture methods can have significantly less environmental burden, but a lot of the meat that is produced these days is through conventional mass production. So surveying some of the literature out there and in life cycle assessment that's been used to quantitatively assess the environmental impact of processes such as meat production, you can see that there's significant potential of cultured beef to reduce the environmental impact in terms of greenhouse gas emissions here and also in land use. And these plots here are generated based on the papers listed here, and they're showing the averages across these different studies. So of course, these numbers could change as processes scale up, but it gives a sense of the potential, I think, to reduce environmental impact. But of course, consumer perceptions are going to be the one of the major driving factors in how much cultured meat actually gets eaten. And this figure here shows synopsis of some of the major barriers in consumer perceptions. This is from a recent review that's coming out, should be any day now, in Trends in Food Science and Technology, together with our collaborator, Janet Tomiyama at UCLA. And from surveys that have been reported in the literature, we identified some of these main barriers. Some people think that it's not going to be as healthy, that it's not natural, disgusting, that it might be too expensive, that it might take jobs away from farmers. So there are various barriers, but the taste is one of the factors that emerged as the top barrier, the greatest concern for consumers, that cultured meat just won't taste as good. It won't have the same flavor as real meat. So this this concern, this barrier, as a scientist and researcher, provides an exciting opportunity because a lot of these concerns can be addressed through science and technology. So what is it that makes meat delicious? Well, certainly the muscle fibers are the foundation of the muscle, but fat is a really important contributor to both flavor, texture, and also mouthfeel. Some of the main flavor components in meat that give it, for example, grassy, fatty, metallic flavors are lipophobic, and so preferentially partition into the intramuscular fat. And you can see by looking at grading systems from around the world, shown here is the US method. On the right here, you can see the Hanwoo Korean beef grading system. And in both cases, you see that the highest quality of meat has the most intramuscular fat or marbling throughout the steak. Of course, and you can see similar features in Kobe beef, the very desirable Kobe beef, and also, of course, in bacon, which has a very high fat content. And you can read more about this, the importance of fat in this wonderful review from the Kaplan Lab, published this year as well in Trends in Food Science and Technology. So a lot of methods, though, to date for engineering muscle focus on the myotubes, which indeed form these muscle fibers that are the foundation of the muscle. So to recap some of those approaches, you have from Mark Post, pioneer in the field, who's shown the expansion of bovine cells on these dextran microcarrier beads that grow quite nicely over days. Other more recent approaches coming from Kip Parker's lab at Harvard show the growth of both bovine and rabbit skeletal muscle on these fibrous scaffolds. There are other approaches that are harnessing the inherent porous structure of plant materials, such as from Glenn Gaudet's group, and shown here decellularized celery from Andrew Pelling's lab, where you can see these C2C12 cells are forming these multinucleated cells that stain with myosin heavy chain, indicative of myotube formation. And yet other approaches use plant materials that have inherent porous structures, such as mycelium, or shown here, textured soy protein that you learned about previously from Tom Benair in this same series, where they're able to grow bovine cells on these textured soy proteins. So all of these approaches are promising in different ways for cultured meat production, but there's still challenges that remain in making marbled cultured meat, and that is namely the challenge of integrating multiple cell types into the cultured meat in a controlled way. So there's been lots of progress, again, from Tom Benair's Nature Food paper, showing that triculture of bovine satellite cells with smooth muscle cells and endothelial cells can enhance differentiation. This is on the textured soy protein scaffolds. And of course, 3D printing methods have a lot of exciting potential to be able to control spatially where you're depositing different cell types and different scaffold materials. So this is ultimately one major challenge, is how you get that spatial patterning. Different cell types also have different media requirements for differentiation, for example. And also different cells can have different mechanical and preferences and need for topographical cues to be able to make their differentiation the most efficient, for example. And finally, scalability is a major criterion that is sort of at the forefront of our thinking in coming up with ways to make marbled cultured meat. So to give an overview of some of those mechanical and topographical cues that are important in myotube differentiation, it's been shown for many years in the literature that aligned fibers can promote differentiation into myotubes. And you can see from this, this earlier work from these human skeletal muscle cells on unaligned versus aligned fibers, you get much more myotube formation on these aligned fibers produced by electrospinning. And work from Adam Engler has also shown that that the stiffness of the underlying matrix is also important for myotube formation as well. You can see here, judging from the alignment of the nuclei, for example, he studied also other features, that this 11 kilopascal stiffness is optimal for myotube differentiation. So taken together, we have a sense that of the optimal kind of physical, mechanical preferences of for myotube differentiation. Less is understood about effects on adipocytes, but we do know that using 3T3 cells as a model system, that on alginate hydrogels, the more compliant or less stiff hydrogels resulted in increased lipid accumulation as opposed to the stiffer matrices. And this is shown here with oil redo staining. But if you recall, the optimal stiffness for myotube differentiation, that was around this 11 kilopascals, so close to this the stiffer condition here. So you can see already how the adipocytes and myocytes have different mechanical preferences for their optimal differentiation and accumulation of lipids. So our approach is as follows, and I say our, I'm talking about Sam and Kathleen and Stephanie here, who've been formed a really effective team, and we've been working together to put together this this approach. But they're the ones who go to the slaughterhouse. So our approach is to create aligned scaffolds that are stiffer, that match the preferences of the myocytes. In parallel, we've been generating more compliant scaffolds for the adipocytes, and then we merge the two scaffolds together, both the muscle tissue and the adipocyte tissue, to make our marbled cultured meat. So I'm first going to talk about the aligned scaffold approach, then the microbead scaffold, and then the integration. So the aligned scaffold was what we started with, and again, there's been methods described in the literature for generating scaffolds that promote myotube differentiation. And one of those is electrospinning, which we know is a scalable method that was one of the first techniques that we started out with to produce these aligned fibers. And so by patterning our collection plate with conductive strips, we're able to tune how aligned those fibers get. You can see shown here in the top panel, these C2C12 cells show increasing differentiation when the fibers are more aligned. And that's shown in this plot down below, just by tuning the distance between the conductive strips, we can generate more aligned fibers. So this was reassuring, and it confirmed what others have seen and reported in the literature. But we ultimately wanted to think towards generating scaffolds that could be compatible with a bioreactor setting. So towards the goal of creating microcarriers that have both tunable mechanics and topology, we relied on our expertise in soft lithography and polydimethyl siloxane microfabrication to generate these grooved substrates that have the C2C12 cells can adhere to, and which we found can also promote their differentiation. And this data here shows these the cells stained for myosin heavy chain and also actin and their nuclei with a hook stain. And you can see on the aligned groove surfaces, they're very highly aligned versus on the flat surface where they have don't show as many aligned patterns. And we've quantified that by doing Fourier transform analysis, which I won't go into the details of, but you can see that that we can get at an orientation index that we quantified for both myotubes, the cytoskeleton based on the F-actin staining, as well as the nucleus, and all the different metrics show that there is a higher degree of alignment both at the cellular level and subcellular level on the aligned grooves. But still, we need to somehow create a more self-contained unit that would be able to be compatible with a bioreactor. So towards that goal, we started generating tiny hydrogel beads, and we did that using water and oil emulsions that you can easily use to similar way you make a vinaigrette or a salad dressing by shaking up water and oil, and if you do that in just the right way with the right surfactants to prevent the droplets from coalescing, you can generate these tiny droplets of water suspended in this continuous phase of oil. And by putting gelatin as well as some microbial transglutaminase into these droplets. They provide the template for forming these hydrogel beads. And you can see here shown on the right some of these gelatin hydrogel beads that are around 100 to 200 microns in diameter. And we can control both the size and the stiffness of these beads. The size we tune just by simple filtration. You can see the size distributions here when we filter, depending on how we filter these beads. We can also tune the stiffness of the beads as well, simply by tuning the weight percent of gelatin that are in the beads. And we can vary the stiffness or Young's modulus of the beads from just around 1 kilopascal up to 40. So around the range that we're exploring for both the adipocyte and myocyte differentiation. So by combining these beads with some of our microfabricated templates, we're able to imprint grooved surfaces onto the microbeads. So now we have these microcarrier particles that have both tunable mechanics and topology. And our results on this are still emerging, and but you can see some of our findings here showing the microcarriers with the patterned surfaces that can support the growth and differentiation of these C2C12 cells, as shown by some of the differentiation markers like MHC. So we're excited to continue working with these microcarriers and better characterize them for supporting the growth and differentiation of our muscle cells. So what about the fat? So for the fat, as I mentioned, the literature suggests that there's a preference for a more compliant, less stiff scaffold. So to address that, we've been, again, using our water and oil emulsions to generate microbeads that could then form the scaffold for adipocytes. And we then culture these in a spinning flask bioreactor. So here are some of the images of our fat cells on the beads, and much to our surprise, but it's not so surprising because cells like to stick to each other. The adipocytes on the beads, these are 3T3L1 cells. They attach to the beads, but then they also start to attach to each other. And so they end up forming these aggregates of adipocytes and gelatin, which is essentially these types of aggregates. So you can see the gelatin beads are faintly green here, so they do get slightly deformed by the cells as these aggregates form. And shown in green here is a lipotoxic dye, so you can see that there is lipid accumulation in these adipocytes on these edible scaffolds. And we find different sizes of aggregates in our spinner flask culture with a large fraction of them over 500 microns in diameter. And again, by filtration, we can tune the size of these as well. And what's interesting is that if they're cultured just in static conditions, we found that they can, the aggregates can grow to the millimeter scale. And this is determined by the size of the well that we're culturing in. So I think there is exciting potential here to achieve the length scale of fat tissue that's needed for marbled cultured meat, which is something you can see by eye. So we've also shown that we can use the scaffolds to support the lipid accumulation in primary rabbit adipocytes as well, not just mouse. And we're currently testing with primary bovine and porcine as well. But I think there's potential here that these scaffolds can support the growth of different animal cell types, because not everyone wants to eat mouse. So finally, how does it all come together? Well, to achieve marbled cultured meat, we take our adipocyte scaffolds and our myotubes on the aligned scaffolds and can integrate them together to form these structurally stable 3D constructs. So, so this is now some confocal data showing you the slices of the from this layered structure where we have primarily the myotubes, primarily the adipocyte scaffolds, and panel C is showing you a projection. And if you look from the side, you can see that the adipocytes and the myotubes are very well integrated with each other. And we're in the process of conducting more detailed mechanical characterizations, but our data is supporting just by qualitative observations that these are cohesive structures that can withstand pipetting, for example. They don't fall apart, and we've been able to do this with both the mouse and also rabbit. Which is perhaps more desirable to eat than the mini mouse meat. But here it is, the mini mouse meat, and again, this has been cultured in small well plates, so the scale seems to be limited by the size of the well at this point. But from our confocal data, we can estimate the density of cells, which is about 10 to the 8th cells per cubic centimeter, which is very close to the target density for meat, which is about 10 to the 8 cells per gram. So, so we think this method has exciting potential to generate cultured marbled meat. So, so that wraps up the talk for today. To summarize, I've shown you proof of concept edible scaffolds for making 3D marbled cultured meat. And also a scaffolding system that can support a lipid accumulation in adipocytes. Finally, a lot of these methods that we've been working with and with the goal of scalability. And so these self-assembling scaffolds should be inherently scalable. Of course, there's still challenges to work on downstream for harvesting to create that spatial structure as in the meat, but that's another challenge that we're working on into the future. But all important steps towards delicious marbled cultured meat. But of course, delicious, as I highlighted at the beginning of the talk, is a really important factor for consumer perceptions and ultimately acceptability and uptake. If no one eats it, you can't reduce the environmental impact, or you can't reap the environmental benefits, potential environmental benefits of cultured meat. But of course, there's other factors as well, in addition to taste. And I come back to the central question of how can we produce now cost-effective, minimum environmental impact cultured meat. So to achieve that goal, I think some of our work really highlights that the intricate relationship between cells and scaffolds and media and the bioreactor setting as well. And all of those factors need to be optimized in an integrated way for maximum efficiency of cell proliferation, for example, differentiation. We know that the stiffness of the matrix can guide the differentiation of stem cells, as work from Adam Engler and others have shown. And so I think harnessing that those physical characteristics of microcarriers has exciting potential to create more efficient systems for cell production. Secondly, we want to make sure that our methods and our source ingredients are able to achieve and reduce environmental impact. And so to that end, we work with our collaborator Deepak Rajagopal to conduct life cycle assessment, which I think done iteratively with our experimental design can help to achieve sustainable cultured meat production and make sure that that we're using source ingredients that are sustainable. And finally, thinking about the uptake and translation of cultured meat into consumers, I think here is where academia and academics have an exciting opportunity to be able to enrich the public understanding of science and provide transparent information on cultured meat. And this is especially urgent given some of the information coming out there that sort of plays on people's fear of chemicals and molecules. So I think with that outlook, the role of scientists could be also in companies as well. Scientists have an important role here to be able to communicate effectively what cultured meat really is, and to promote food literacy from an educated perspective. So on that note, I'd just like to thank you all for attending, no matter what time of day or night it might be for you. And again, you should come and visit us in Los Angeles. Now is not the best time, but soon the smoke and viruses will hopefully subside, and come and visit our group. And thank you again to GFI and all of our other support for making this research happen. So I'm at this point happy to take questions. Thank you.
Awesome. Thank you so much, Amy. That was such a fantastic seminar, and the chat was blowing up with lots of people saying how inspiring and promising your research is looking. So I think we're all excited to follow its progress. We do have a number of photos, and there are now people in the chat saying that they will definitely come visit you.
Right, great.
Good. There are a number of questions here in the Q&A section. For those of you who aren't familiar with this kind of Zoom webinar format and have questions for Amy, you could go ahead to the Q&A section and input your questions, and we will get to them in the next half hour. All right, so let's take our very first question from Simon Kahan over in Washington, who runs the Cultivated Meat Modeling Consortium. His question for you, Amy, is how does the test compare with atomic force microscopy, where elasticity of cells could be determined? In our modeling efforts, we need to know not just how deformable a cell is, but also whether or not it bounces back.
Right. And so the test, I'm assuming, is, Simon, you're referring to the filtration method that we've developed. But I can speak to both. So our filtration methods, we find we have both this bulk filtration, but also a microfluidic method that we've developed for quantifying cell elastic modulus as well, that allows us to visualize individual cells deforming through microfluidic constrictions, and that enables us to also back out an elasticity for cells. So we found that there's very good agreement in between atomic force microscopy measurements and our fluidic methods for measuring cell deformability. But in some cases, the depth of the penetration of the mechanical measurement differs. So we do see in some contexts different results. But in terms of the bouncing back, the relaxation of the cells, atomic force microscopy is one way to be able to measure that now. There's more advanced methods for measuring cell relaxation that companies as well as researchers, like I know Todd Sulchak, for example, is working on that. And we've also been working actively on measuring cell relaxation in our microfluidic method. So we can visualize cells as they squeeze through the narrow gap and then pop out again, and we can measure the time scale of that relaxation. So definitely would be great to talk more about how our experiments and modeling efforts could coincide.
Awesome. Thank you, Amy. It's going to get weird with me saying my own name so many times.
Right.
I'm getting confused. Okay, we have a question here from an anonymous attendee. Do you think suspension cell aggregates could work as an approach for muscle and fat differentiation?
And so by this, suspension cell aggregates, I'm assuming no scaffolding involved. That could be approached. We haven't looked extensively into the muscle differentiation with of just the cell aggregates, but certainly with the fat cells, we do see that that there is some uh, that could be an approach of just the cell aggregates, but we're still in the early stages of fully characterizing the effects of the different microcarrier physical properties versus just cell aggregates as well. So, so it's a good question.
Great, thank you. Another question here from Oscar. When translating this to feasible applications, I wonder if there's a place for combining culture methods with some level of differentiation to make parts that make up a whole steak.
Mm-hmm. Absolutely, and that seems to be the lowest hanging fruit that we're pursuing right now, where we can generate the sort of separate components of the muscle and the adipocytes, and then fuse them together. But I do think that in translation, that could be an effective strategy as well. Of course, with higher throughput methods for harvesting and spatial patterning. But given the different media requirements for optimal differentiation, I think that that might be a feasible path in the future.
Awesome, thank you. All right, we have a growing list of questions here. One question from Anna. Has your group ever looked into the stiffness preference in other adipocyte cell types, primarily cells from other animal sources, for differentiation? She's curious because their lab works in extracellular matrix and adipocyte thermogenesis, and they've seen a significant difference in matrix stiffness and uncoupling protein 1 expression in primary differentiated adipocytes?
This is an excellent question, and we are just starting to work on primary rabbit cells as well, rabbit adipocytes right now. So so we've so far looked at the 3T3 cells, and but this is a great question because it's the ultimate translation to primary cells is of observations in model cell systems is definitely something that needs to be validated. So so that's in progress.
Wonderful, thank you. There are 2 questions here about fatty acid profiles, and I'll just kind of combine them. So I think the questions are, what determines fatty acid profiles? How do you control them? And how do they relate to health and flavor and oxidation?
This is a great question because, yeah, as you point out, those fats are really important for both health and flavor. And if we could tune those, we could perhaps generate a more healthful fatty or flavorful fatty acid profile. So we haven't gotten into that stage of our research yet, but definitely have some experiments planned to better understand how those physical cues, as well as different soluble factors in the microenvironment, can impact fatty acid composition in cells as well. So we have, for example, a screening resource core facility at UCLA that has libraries of natural compounds that we're excited to be able to screen, to be able to identify factors that could, shift the fatty acid profile to be more beneficial for cultured meat applications. A lot of small molecules, natural compounds out there have been investigated for the opposite effect for obesity, diabetes treatment. So, so I think better understanding how both these physical and mechanical cues come together to regulate fatty acid composition is an exciting direction.
Fantastic. Here's a question from Theodora. Hi, Amy, really wonderful talk. Their question is, how do you deposit the adipocyte aggregates onto the myotubes on gelatin, and in which medium do you grow these composites?
So currently we've been depositing the adipocyte aggregates onto the myotubes, just manually stacking, but we have some more automated procedures that we're working on, sort of using our expertise in fluids and fluid manipulations. And then the second part was the medium that we used. We sort of developed we sort of screened various combinations of media that support both the adipocytes and the myotubes to arrive at a medium that was supportive of both the cell types. But I should mention too, I forgot to mention that in the talk, that the adhesion happens rather quickly, that for the mouse around 3 hours, for the rabbit mini meat, marbled meat, around 6 hours. So the cells don't necessarily have to be cultured in the same medium together for a long period of time to be able to form a cohesive meat structure.
Yeah, I have to say mini meat might be my favorite term that's been coined in the course of our scientific seminar series. Here we have a couple questions about like animal-free components. So one question is about whether the gelatin you used in this study was produced through fermentation or whether it was animal sourced, and whether you think we could use a plant-based gelatin.
Right. Yeah, so this was animal source gelatin, and we had initially started out using pectin and alginated scaffolds, which we've also used to make the microbeads scaffolds as well. But of course, then we need additional steps to functionalize, to get the cells to the mammalian cells to adhere. And so given the developments in microbial produced gelatin, we opted for gelatin with the end goal in mind that we could identify a lower environmental impact source of gelatin, or plant-based gelatin, in the future. So I think that was there another part of that question? Was that?
Sorry, I was on mute. I was saying that I think that was a really comprehensive answer.
Okay. Okay.
The kind of second part of the question is around serum-free media.
Right. Okay.
Yeah.
So yeah, similarly with the medium, we've been using, we have not been using plant-based medium, but with the goal that that for proof of concept, this is what we've done, and the findings can then be translated to I think in the end you need to do more screening of different media scaffold cell combinations to optimize that for animal-free cultured meat, or using scaffolds and media.
Yeah, thank you. There's certainly lots of parallel research efforts happening in the cell culture media space and the scaffolding space. Cool. So there's a question here from John. Are the marbled meat constructs for cultivated muscle and fat layered together, or are they mixed together in some other way?
They are layered together. Yeah, and that's currently how we're doing it. It's the layering.
Great, thank you. Only 21 questions left. You're doing great. The question here from Lei, thank you for a wonderful talk. They're wondering what cell source you would be using to obtain myocytes for the final cultivated meat product.
Right. So as I mentioned, Sam and Kathleen and Stephanie, our mini meat group, has been making trips to the slaughterhouse. So we're working on isolating satellite muscle cells currently that we would be able to show, but again, the goal is to show proof of concept, and so that would be the source that we would be using for our final cultured meat product. Or final cultured meat wouldn't be a product, but yeah. Right. But I think these the methods could be generally applicable to different types of cells from different sources.
Yeah, thank you. A question here from Christoph Shanter from Kip Parker's lab, actually. Are the patterned beads flattened almost two-dimensionally to imprint the pattern?
So they're not quite 2D, but we're still working on sort of tuning the final shape of the beads, but they could be sort of flattened into 2D construct, but they don't necessarily have to be.
Awesome, thank you. And we have a question here from Varsha. How do you think the inclusion of the gelatin microgels affects the texture of the meat? Do you have plans to remove those beads as the cells grow?
This is a great question, and we don't know the answer to this yet. We anticipate that there will be some degradation of the gelatin beads over time as the cells grow, but they are also cross-linked with the transcontaminase. So we still don't know exactly how those are going to affect the final texture, but upon cooking, we know that gelatin is a really important contributor to unctuous mouthfeel that's desirable in meat products. So it could be a benefit. So I think that's a great question. And yeah, we don't have plans to remove the beads, but I think figuring out how the stiffness of the beads, the amount of gelatin that's in there will affect the ultimate final texture of the cultured meat.
Great. We have another question here from Oscar. Is there a theoretical size limit when the nutrient transport rate would be limiting? So
Right.
Is it possible to envision including vasculature or a functionally analog perfusion method as part of the matrix to remove the limitation?
Right. Central question to all of our work in cultured meat. So I think this method of sort of merging, fusing multiple cell constructs could allow for endothelial cells, for example, to be engineered into the cultured meat constructs. But I think also our approach could sort of get around some of the size limitations. You know, typically like a couple hundred microns would be like the size limitation over which you start to get cell death due to limited nutrient exchange. But a couple things to note about our scaffolds. One is that others have shown using fused microbeads, such as Dino DiCarlo and Tatiana Segura, that these are macroporous, and so there is enhanced diffusion through these scaffolds with cells. Those were formulated, fabricated in a slightly different way, where those beads were cross-linked together before the cells were seeded. So we still don't know exactly what the porosity of our constructs are. And because we have such high cell densities of 10 to the 8 cells per cubic centimeter, clearly the diffusion will be lower, I think, through our scaffolds. But I think one promising thing to mention is that the scaffolds fuse together on the timescale of hours. So you might not actually need to culture a thick cut of meat for that long after it's fused together. So you could imagine bringing together these smaller aggregates, fusing them, and then just chilling your meat, and it's done and off to be processed. And so you wouldn't actually need to have exchange of nutrients and for over long time scales. So that's a long-winded answer to your question, but it's a very good one.
Yeah, that was super fascinating. I learned a lot. Thank you. Taras has a question about how you combine the two-dimensional planes for PDMS, grooved surface, with the three-dimensional spherical shape of the gelatin microbeads.
So the PDMS grooved surface, the 2D one, we're not actually pursuing that as a way to integrate that with the 3D gelatin microbeads, but we do use the 2D surface as a way to imprint the topology onto the microbeads.
Awesome. It looks like that second question is about whether 3T3 adipocytes can be grown without microcarriers.
So that came up a little bit before, and we're still sort of in the early stages of characterizing how the 3T3 cells can be grown without microcarriers, but they have been. It is possible, yes.
Awesome. Okay, here's a question on a different note about the other programs that you're involved in. Nicholas is wondering whether you can speak a little bit more about Science and Food and Eat Well as the two programs that you work in. Yeah.
Great. Happy to. So Science and Food, as I mentioned, was based on this course that I teach every spring at UCLA, where I have the opportunity to work together with chefs and farmers and food visionaries to provide real life examples of the aspects of science in food and cooking. We also, from these efforts, spawned a public event series where we bring together experts, again, spanning science and culinary arts and authors and economists, people that are thinking about food from all different perspectives. And so some of our recent events, for example, included an event on the future of food in extreme environments, where we had a chef thinking about extra or planetary travel with engineer working on developing greenhouses that NASA can use for their space stations, and a food technologist who worked at NASA for many years. So the idea is to bring together people from different backgrounds to bring a sort of scientific dialogue to timely topics on related to food. And then we also have some undergraduates working on curriculum development for younger audiences, like high schools, for example, that translate some of the science and food curriculum that I teach at the undergraduate level for high schools. So the other question was about the Eat Well group that I co-lead. So the Healthy Campus Initiative, Summer Healthy Campus Initiative at UCLA is a philanthropist backed by Jane and Terry Summel effort to make UCLA the healthiest campus in the country, one of the healthiest campuses in the world. So there are various groups that focus on improving the built environment, making more bike paths, for example, our mental well-being, providing resources for meditation and psychological well-being, and food is another one of those pods that I co-lead together with operations expert in UCLA housing and hospitality. So we have we put on various have various efforts to promote food literacy. We've also worked on this LAUSD, LA Unified School District curriculum. We do a lot of outreach and on UCLA campus as well, raising awareness of the environmental impacts of food waste. Those are just a couple of topics we've covered in recent years. This year, we'll have a focus on social justice in the food system. So we have these kinds of events and generate educational materials, and also spawn research activities as well. So people have been studying, for example, in the School of Public Health, how the uptake of Impossible Burgers and UCLA dining has gone, and looking at the effect of specific informational campaigns. So we have this living laboratory that's a pretty cool way to be able to do live studies. So hopefully that answers your question, but let me know, send me an email if you want any more information.
Thank you so much, Amy. Yeah, I've heard really great things about the public lecture series that Amy runs as part of this Science and Food program, and I would highly encourage everyone to check it out if you're excited about the broader picture of food tech and the way that it kind of intersects with all of these interesting issues that Amy just mentioned. So very cool. We have 16 questions left and only 11 minutes left on the clock. So I'll kind of pick pick some of the ones here from the top, and Amy, if there are any that you're particularly excited about answering, you can feel free to jump in at any point. Here's a question from Varsha. Could you explain your integration process? Is it centrifugation based? Do these material platforms, or are they amenable to 3D printing?
Great. Yeah, so I mentioned previously that our proof of concept has been using this manual integration process, but certainly we're considering centrifugation and also filtration technologies to be able to automate the integration process. And certainly these materials are amenable to 3D printing. We've been trying to find more scalable, inherently scalable ways to generate our scaffolds and the tissue and the integration, just to avoid or to provide a more inherently scalable system. Yeah, so we haven't explored 3D printing ourselves, but it's a very powerful approach.
Thank you. Here's a question from Shira from the Technion. Can you please elaborate about soft lithography? Does it not harm the hydrogel scaffold?
So the soft lithography is a way that we can produce structures with micron scale features, such as these templates to make these grooved hydrogel structures. So we use it kind of as a mold for the curing the hydrogel. So we can pour the polymers in, and then they form a gel in the shape of our mold.
Wonderful. Here's a question about asking if you can predict that there might be changes to the growth rate of adipocytes and myocytes during co-culturing or the integration of scaffolds as compared to monoculturing.
Certainly that's a major challenge in the co-culture. And our approach, we've been doing the proliferation separately of each of the cell lines. So the co-culture is really just when the cells are co-culturing is when they're fusing together on the timescale of hours. So the proliferation happens previously.
Great, thank you. This is a related question to some of the questions around animal-free materials, but it's a little bit broader than that, so I'll see if you have anything else to supplement here. Are there components in your current methodology or formulation that would need to be removed or filtered out in the end product? And if yes, what methodologies would you explore to do that?
That's a great question. I mean, currently we're using filtration just to tune the size of our of the components, the desirable components. So that is one thing that comes to mind, but I'm not sure how critical that would be for the end product. But yeah, it's an interesting question.
Thank you. All right, here's a question from Stacy Scalur at GFI Europe. When you assembled your constructs, did you see any evidence of myotubes that were able to bridge across and connect between the patterned microcarriers?
We haven't been able to look in that level of detail just yet, but those experiments are in progress, and it's a great question, Stacy. Thank you.
Awesome. This question is interesting. How feasible do you think it would be to predetermine the tissue content and percentage in the potential cultivated steak?
That is the vision. Yeah. So I think by tuning the size of the adipose tissue aggregates, and of course that's also requires some higher throughput way for patterning, spatial patterning, which is, yeah, work that's in progress. So, but that is absolutely the goal, that you could pattern, and not just for steak, but different types of patterns for salmon, for example, or different bacon, different forms of marbling. So, so yeah, definitely that question is aligned with our vision.
Awesome, thank you. There's a question here from Gonzalo. You may have answered it, but he asks, what kind of functionalization were you performing on the pectin or alginate microbeads that you were previously using?
We did an RGD functionalization, but we very quickly switched over to the gelatin just for removing and streamlining the process.
Thank you. Um, and there are some questions here from Jake Reese. Did you use mature adipocytes or fibroadipogenic progenitors to conjugate your microcarriers?
So we use the 3T3 cells and then differentiated them, and we're working right now with the primary rabbit adipocytes.
All right, thank you. Looks like we have 6 questions left. Are any of them calling out to you?
So I guess there's one question here about from Erica about the fibril scaffolds. What are they made out of? And we've been using gelatin for our scaffolds. Here's a question about what advice I may have for a recent university graduate that wants to work in cultivated meat but doesn't have any lab research experience. I think attending seminars such as these, getting an overview of the field is a great way to start. GFI has some awesome resources for doing that, and I think reaching out to labs or companies even to get your foot in the door and start gaining experience is a great way to start. And maybe you don't start right away in doing the research, but can sort of work your way in there. And maybe there's another skill that you have that's not research based, but can help to advance the industry as well. I think we need people with all different skill sets and backgrounds.
Yeah, thank you, Amy. That's a great answer to that question. I think this is such a relatively uncharted field that people are really, really excited to be helping each other. And I love Amy's advice to kind of reach out to the labs that you're interested in working for and see if they're open to mentoring you and helping you get some of that experience. People in general in this field seem to be really open to that kind of mentorship and collaboration. Awesome. So with 3 minutes left, I just want to kind of thank you again, Amy, for this really fantastic seminar. And I want to give you a chance to give any closing thoughts that you might want to leave our community with today.
Thank you. Well, thank you for this opportunity. I think it's a really exciting time, and it's really ripe for discussion and conversations across disciplines. And of course, within, cell biology, bioengineering, materials science, but more broadly with behavioral scientists and environmental scientists. I think that there are a lot of problems and questions that really span disciplines and require expertise on all fronts. And yeah, as I mentioned also in the closing of my talk, I think that that as a community, we also have an important role in communicating with the public and potential consumers as well. That may have doubts and skepticisms about cultured meat. Maybe that's lingering from memories of GMOs, for example. But I think we have the opportunity also to provide and provide transparent information and address some of those concerns up front as cultured meat is going to come to market.
Yeah, that's great. Thank you so much, Amy. And I think you're absolutely right. There's an opportunity now to start engaging the consumers of these future food products so that, yeah, we're making sure that we're appeasing any of their potential concerns down the road. This was such a fantastic seminar. I'm so grateful to you, and
thank you.
So grateful to all of our attendees who have joined us from all around the world to learn from you today. Thank you again for being a part of this community. Next month, like I mentioned at the start of this seminar, we will be joined by Dr. William Chen from NTU, and he'll be talking about how we can utilize agricultural waste streams from grains as kind of a microbial fermentation fuel. And that's going to be pretty important because we need to be very resourceful if we're going to build a more sustainable food system and utilize all of these waste streams as potential inputs for alternative protein products. So thank you again, everyone, for joining us today. Thank you, Amy. You're wonderful. And I hope everyone has a fantastic rest of their day.